The physiology of the sensory organs refers to how these specialized organs function to detect and process different types of stimuli from the environment, such as light, sound, smell, taste, and touch, convert them into electrical signals, neural impulses, and transmit them to the brain for interpretation. TAC Introduction. The human body has 5 primary sensory organs: eyes, vision; 2 ears, hearing and balance; 3 nose, smell; 4 tongue, taste; 5 skin, touch, including pressure, temperature, and pain. Each organ contains specialized sensory receptors that respond to specific stimuli. Number 1, eye, vision. Sensory receptor, photoreceptors, rods and cones in the retina. Stimulus, light, photons. Physiological process. Light enters through the cornea and lens, focusing onto the retina. The retina contains rods for dim light, black and white vision, and cones for bright light and color vision. Light activates photopigments, triggering a phototransduction cascade. This produces electrical signals that are processed by retinal neurons and transmitted via the optic nerve to the visual cortex of the brain. Number 2, hearing and balance. Sensory receptors. Hair cells, mechanoreceptors, in the cochlea and vestibular system. Stimulus, sound waves for hearing, and head movement for balance. Physiological process. Hearing. Sound waves travel through the auditory canal, vibrating the tympanic membrane eardrum. Vibrations are transmitted via ossicles, malleus, incus, stapes, to the cochlea. Inside the cochlea, vibrations move the basilar membrane, stimulating hair cells in the organ of corti. Hair cell movement generates nerve impulses sent via the auditory nerve to the auditory cortex. Balance, equilibrium. The vestibular apparatus, semicircular canals, utricle, and saccule detects head movements and position. Fluid movement within canals bends hair cells, sending signals to the brain about orientation and motion. 3. Nose, olfaction, smell. Sensory receptor, olfactory receptors, chemoreceptors, in the olfactory epithelium. Stimulus, chemical molecules in the air. Physiological process. Odor molecules dissolve in mucus of the nasal cavity. They bind to olfactory receptor neurons, triggering a signal transduction pathway. This creates action potentials that travel through the olfactory nerve to the olfactory bulb and then to the olfactory cortex for interpretation. Number 4, tongue, gustation, taste. Sensory receptors, taste buds, chemoreceptors, on papillae of the tongue. Stimulus, chemical substances in food and drink. Physiological process. Food molecules dissolve in saliva and interact with taste receptors. Each taste bud responds to one or more of the 5 basic tastes: sweet, sour, salty, bitter, umami. Activation of receptors generates electrical signals transmitted via facial, lesson, glossopharyngeal, and vagus X nerves to the gustatory cortex. Number skin, touch and somatosensation. Sensory receptors, mechanoreceptors for pressure and vibration, thermoreceptors for temperature, nociceptors for pain. Stimulus, mechanical pressure, temperature changes, pain-causing stimuli. Physiological process, stimuli activate specific receptors in the skin. Signals travel via afferent sensory neurons to the spinal cord and then to the somatosensory cortex of the brain. The brain interprets the location, intensity, and nature of the stimulus. Integration in the central nervous system, CNS. All sensory signals except smell synapse in the thalamus before being relayed to specific areas in the cerebral cortex for perception. Sensory integration allows the brain to create a comprehensive experience of the environment. Conclusion. The sensory organs convert physical or chemical stimuli into electrical signals through specialized receptors. These signals are then interpreted by the brain, allowing us to perceive and react to our surroundings. Each sense has unique mechanisms, but all share the fundamental process of stimulus detection, signal transduction, neural transmission, brain interpretation. Here's a clear explanation of the functions of the sensory organs without using a table format. Functions of the sensory organs. 1. Eyes, vision. The eyes allow us to see by detecting light and converting it into electrical signals. They help in perceiving shapes, colors, distances, and motion. Eyes also support depth perception, visual learning, and coordination of movement. Light exposure through the eyes helps regulate the body's internal clock, circadian rhythm. 2. Ears, hearing and balance. The ears detect sound waves and convert them into nerve impulses, allowing us to hear. They help us understand speech, enjoy music, and respond to environmental sounds, like alarms or voices. The inner ear, vestibular system, plays a crucial role in maintaining balance, posture, and spatial orientation. 3. Nose, smell, olfaction. The nose detects airborne chemical molecules, allowing us to perceive different odors. It plays a key role in the sense of smell, which is linked closely with taste. The sense of smell also alerts us to dangers like smoke, gas leaks, or spoiled food. 4. Tongue, taste gustation. The tongue senses different tastes: sweet, sour, salty, bitter, and umami. It helps identify nutritious food and detect harmful or spoiled substances. It also plays a role in chewing, swallowing, and forming words during speech. 5. Skin, touch, and somatosensation. The skin contains receptors for touch, pressure, temperature, and pain. It allows us to feel sensations like softness, heat, cold, or sharpness. Touch helps us interact with our environment, protects us from injury, and plays a role in emotional bonding. Conclusion. Each sensory organ performs a specialized function essential for interacting with the world. Together, they allow us to perceive, interpret, and respond to our surroundings, ensuring safety, survival, communication, and enjoyment of life. Physiology of skin. The skin is the body's largest organ and plays a vital role in protection, sensation, thermoregulation, excretion, and vitamin D synthesis. Its physiology refers to how the skin functions at the cellular and systemic level to maintain homeostasis and interact with the environment. 1. Structure of the skin, basic overview. The skin has three main layers. Epidermis, outermost layer, contains keratinocytes, melanocytes, and Langerhans cells. Dermis, middle layer rich in collagen, elastic fibers, blood vessels, nerves, and glands. Hypodermis, subcutaneous tissue, deepest layer, composed mainly of fat and connective tissue. 2. Major physiological functions of the skin. A. Protection, physical barrier against pathogens, UV radiation, and harmful chemicals. Keratin in the epidermis provides mechanical strength. Melanin, produced by melanocytes, protects against UV light. Langerhans cells in the epidermis detect and alert the immune system to foreign invaders. B. Sensation. The skin contains specialized sensory receptors for touch, via Meissner's corpuscles and Merkel cells, pressure, via Pacinian corpuscles, temperature, via thermoreceptors, pain, via nociceptors. These receptors send signals through peripheral nerves to the central nervous system, allowing perception of environmental stimuli. C. Thermoregulation. Sweat glands secrete sweat, which cools the body through evaporation. Blood vessels in the dermis dilate to release heat, vasodilation, constrict to conserve heat, vasoconstriction. Adipose tissue in the hypodermis provides insulation. D. Excretion. Sweat glands excrete water, salts, and small amounts of urea. This helps remove waste and regulate electrolyte balance. E. Vitamin D synthesis. The skin synthesizes vitamin D3, cholecalciferol, when exposed to UVB radiation. This precursor is later converted in the liver and kidneys into active calcitriol, essential for calcium absorption and bone health. F. Immune function. Langerhans cells in the epidermis capture and present antigens to immune cells. The skin acts as the first line of defense in the innate immune system. G. Wound healing. The skin heals in 4 key phases. 1. Hemostasis, blood clot formation. 2. Inflammation, immune cell recruitment. 3. Proliferation, cell division and new tissue formation. 4. Remodeling, collagen restructuring, and scar formation. The skin is a complex organ with vital physiological functions including protection, sensation, regulation, excretion, and immunity. Its unique structure enables it to serve as a dynamic barrier and a sensory interface between the body and the external environment. Physiology of vision. The physiology of vision refers to the process by which the eyes detect light and convert it into electrical signals that are interpreted by the brain to produce the sense of sight. 1. Pathway of vision. The process of vision involves the following key steps. A. Light entry into the eye. Light enters the eye through the cornea, the transparent front part of the eye. It then passes through the aqueous humor, pupil, lens, and vitreous humor before reaching the retina. B. Refraction of light. As light travels through the cornea and lens, it is refracted bent to focus the image on the retina, especially on the fovea centralis, the point of sharpest vision. 2. Retina and photoreceptor function. A. Photoreceptors, rods and cones. The retina contains two types of photoreceptors, rods, sensitive to dim light, responsible for black and white vision and night vision. Cones, responsible for color vision and visual acuity, function in bright light. Types of cones, sensitive to red, green, and blue wavelengths. B. Phototransduction. Light activates photopigments, e.g., rhodopsin in rods. This triggers a biochemical cascade resulting in the generation of electrical signals, action potentials. Photoreceptors transmit signals to bipolar cells, then to ganglion cells. 3. Signal transmission to the brain. Ganglion cells, axons, converge to form the optic nerve. The optic nerve carries visual information to the optic chiasm, where fibers from the nasal side of each retina cross over. Signals continue to the lateral geniculate nucleus, LGN, of the thalamus. From there, visual signals are sent to the visual cortex, occipital lobe of the brain. 4. Interpretation in the brain. The visual cortex processes shape, color, depth, movement, position. This allows us to form a clear, 3D, colored, and meaningful visual perception of our surroundings. Other physiological aspects. A. Visual accommodation. The lens changes shape via ciliary muscles to focus on near or distant objects. B. Pupillary reflex. The iris controls the size of the pupil to regulate light entry. Constriction in bright light, miosis. Dilation in dim light, mydriasis. C. Color vision. Depends on stimulation of different cones for red, green, and blue. Color blindness results from defective or missing cones. Conclusion. Vision is a highly coordinated process involving 1. Light detection by the eye, 2. Signal conversion by photoreceptors, 3. Transmission through neural pathways, 4. Image processing in the brain. This system allows us to perceive and interpret the world in rich detail, color, and depth. Let me know if you'd like a labeled diagram or mnemonic. Physiology of hearing. The physiology of hearing involves the detection, conversion, and interpretation of sound waves. This process allows us to perceive and respond to sounds in our environment. 1. Sound wave collection. Sound waves are vibrations in the air. These waves are collected by the pinna outer ear and directed into the external auditory canal. They strike the tympanic membrane eardrum, causing it to vibrate. Vibration transmission, middle ear. Vibrations from the eardrum are transmitted to 3 small bones in the middle ear: malleus, hammer; incus, anvil; stapes, stirrup. These ossicles amplify the vibrations and pass them to the oval window, a membrane at the entrance of the inner ear, cochlea. 3. Cochlear function, inner ear. The cochlea is a spiral-shaped, fluid-filled structure in the inner ear. The vibration of the oval window creates waves in the cochlear fluid. These waves move the basilar membrane inside the cochlea. Hair cells and sound transduction. On the basilar membrane is the organ of Corti, which contains hair cells, mechanoreceptors. As the basilar membrane moves, hair cells bend against the tectorial membrane. This bending opens ion channels, creating electrical signals. 4. Transmission to the brain. Electrical signals from the hair cells are sent via the cochlear nerve, part of the vestibulocochlear nerve, cranial nerve 8. These signals travel to the cochlear nuclei in the brainstem, inferior colliculus, medial geniculate nucleus, MGN, of the thalamus, auditory cortex in the temporal lobe. Here the brain interprets the sound, pitch, loudness, and location. 5. Pitch and loudness perception. Pitch, frequency, determined by which hair cells are activated. High-frequency sounds stimulate hair cells near the base of the cochlea. Low-frequency sounds stimulate those near the apex. Loudness, intensity, determined by how strongly the hair cells are stimulated. 6. Roll and balance vestibular function. Although not directly related to hearing, the inner ear also contains semicircular canals, detect rotational movement. Utricle and saccule detect linear acceleration and gravity. These structures help maintain balance and spatial orientation. Conclusion. Hearing involves 1. Collecting sound waves, 2. Converting them into mechanical vibrations, 3. Transforming them into fluid waves in the cochlea, 4. Stimulating hair cells to generate electrical signals, 5. Transmitting and interpreting signals in the brain. Physiology of taste, gustation. The physiology of taste refers to how the body detects chemical substances in food and drinks and converts them into nerve signals that are interpreted by the brain as taste. It is one of the 5 special senses and is closely linked to smell and saliva. No. 1. Taste receptors and taste buds. The tongue, soft palate, pharynx, and epiglottis contain taste buds. Taste buds are made up of gustatory receptor cells, which have microvilli, taste hairs, that project through taste pores. These receptors are stimulated by chemicals dissolved in saliva. 2. 5 primary tastes. Humans can detect 5 basic tastes. 1. Sweet, usually indicates energy-rich nutrients, sugars. 2. Sour, detects acids, H-brie. Salty, detects sodium, NE5 ions. 4. Bitter, often a warning signal for toxins. Umami, detects amino acids, especially glutamate savory. 3. Process of taste perception. A. Stimulation. Food molecules are dissolved by saliva. These dissolved chemicals interact with specific receptors on the gustatory cells within taste buds. B. Signal transduction. Depending on the type of taste, different mechanisms are activated. Salty and sour tastes involve ion channels. Sweet, bitter, and umami tastes use G-protein coupled receptors, GPCRs. The interaction leads to depolarization of the taste cell and release of neurotransmitters. C. Nerve transmission. Neurotransmitters stimulate sensory neurons connected to the taste buds. Signals are carried by cranial nerves, facial nerve, 7, front 2/3 of tongue, glossopharyngeal nerve, back 1/3 of tongue, vagus nerve X, epiglottis, and throat. Number 4. Interpretation in the brain. The sensory information is first processed in the gustatory nucleus of the medulla oblongata. It is then relayed to the thalamus and finally to the gustatory cortex in the insula and frontal lobe of the brain. Here, the brain interprets the type and intensity of taste. Number 5. Factors influencing taste. Smell enhances flavor perception, e.g., food tastes bland when you have a blocked nose. Texture and temperature affect how taste is experienced. Age and health. Taste sensitivity decreases with age or illness. Medications can alter taste sensation. Conclusion. The physiology of taste involves 1. Detection of chemicals by taste buds, 2. Signal transduction into nerve impulses, 3. Transmission to the brain, 4. Interpretation into recognizable tastes. Thank you. Have a nice day.